Boa tarde, pessoal.
Privacy.
Bom, a gente vai dar um início aqui enquanto eles terminam de ajeitar ali a professora Pamela. Para a professora Patrícia Morelato, aqui da Biodiversidade, da Unesp de Rio Claro, ela vai fazer uma abertura rápida aqui para a gente. Então, nosso primeiro, bem-vindos e obrigado pela presença de vocês. A gente vai fazer uma pequena abertura, né, a professora Patrícia. E nesse primeiro dia do evento, né, então amanhã contamos com a presença de vocês para continuar o evento, tá? Mas é isso. A Patrícia vai falar um pouquinho para a gente, uma abertura breve, e depois a gente vai ouvir a palestra da professora Pamela.
Oi, pessoal, boa tarde. Primeiro de tudo, eu queria agradecer todo o pessoal que fez essa organização, começando com a Mi, que assumiu a liderança, e com todo o grupo de Natural History, que ajudou e vem organizando isso, que é o nosso primeiro evento para congregar todo mundo que está participando do CEPID. Depois a gente vai ter nas outras áreas também o CEPID. Amanhã vocês vão saber como é que é o CEPID, os 4 pacotes que ele tem, depois a difusão e a inovação. Mas hoje, então, a gente começou com esse que é um dos workpacks que também se conecta com todos os que a gente trabalha. Então, eu agradeço muito, agradeço toda a equipe, todo mundo que se envolveu nisso desde o começo, fizeram muitas discussões. E a organização foi muito mais do que a gente imaginava, né? São mais de quase 150 pessoas inscritas, assistindo a gente. Então, ou seja, a gente tem um grupo muito legal e vamos fazer muita coisa bacana e contamos com todos vocês, né? Então, isso é muito importante. E hoje, então, só essa abertura. Lembrando vocês que agora o professor Vitor vai apresentar a professora Pamela, que eu agradeço muito também. And thank you very much, Pen, for coming here and give the talk. Vitor is going to introduce you, but I would like to thank you. I'm Patrícia Morelato, and thank you very much. E vou fazer lembrar vocês que à tarde, depois, hoje, tem essa abertura e depois vamos tomar um chopp juntos. Né? Como é que é o nome? Sempre esqueço o nome. Heisten Pub. Tá legal? Conto com vocês nessa outra atividade essencial, que sempre é lá que surgem mais ideias ainda. Tá bom? Então, vou passar para o professor Vitor, que também é o coordenador é o outro pesquisador principal do projeto e que coordena a internacionalização e faz de tudo comigo aqui também. Muito obrigada, Vitor, e todos os pesquisadores principais, associados, pós-docs, de graduação ou pós-graduação, todos vocês fazem a gente, que hoje a minha amiga falou, a Tereza, que está com o coração quente de ver como as coisas, mas não tinha como ficar frio nesse calor, mas está mais aquecido ainda, tá? Muito obrigada.
Obrigado, Patrícia. Bom, boa noite a todos. Vamos dar início, então, fazendo a apresentação da professora Pamela. Hi, Pam. Well, we are thrilled to introduce our distinguished speaker for the first Cbioclima workshop delving into the theme Natural History from Genes to Ecosystems. The presenter, Professor Pamela Soltis, will lead a captivating discussion on repurposing herbarium specimens for studies of climate change. Professor Pamela Soltis from the University of Florida is a prominent figure in the scientific community, making fundamental contributions to understand phylogenetic relationships of flowering plants within the framework of a multifaceted project, the Tree of Life. Her work encompasses unraveling the genetic basis of floral development and emphasizing the crucial importance of polyploidy in plant evolution. As a distinguished member of the National Academy of Sciences and the American Academy of Arts and Sciences, Professor Soltis brings a unique and broad perspective to the discussion on climate change and its impact on plants. Her unparalleled expertise in the study of plant diversity, focusing on the origin of evolution of flowering plants, plant genome evolution, and conservation genetics, positions her as a visionary leader in the field. As a director of the University of Florida Biodiversity Institute and a member of the University of Florida Genetics Institute, Professor Pamela Soltis plays a crucial role in shaping the next generation of scientists and promoting interdisciplinary research. In over 400 peer-reviewed articles, her diverse laboratory is a hub of innovation and discovery. Thank you very much, Professor Pamela, and we are very pleased to bring you here and your presence. Thank you.
Well, thank you so much for the invitation to participate in your workshop. And I am just sad that I am not able to be there in person. I hope that the workshop goes well and that you have a great couple of days exploring topics related to biodiversity and climate change. I'm also wondering, I guess I want to make sure everyone can hear me okay. And also if I should change my screen sharing so that you can see, I think you're not seeing the full screen, is that right? Should I change just a second? Well, no, if that's like that, I can't see you guys. Is that better though? Okay. All right. Well, I will just go from there without notes, etc. So I'm also very sorry for the delay getting started. My computer just needed to be restarted apparently. So I think we have everything set now. So again, I'm very happy to be with you today and to be helping you proceed through this workshop. So as botanists, ecologists, and evolutionary biologists, we are all familiar with herbarium specimens and other natural history collections. But what we may not always think about is how these collections can aid us in various types of research. And so what I would like to do today is share with you some of the ways that we have been using both data and images of herbarium specimens to address questions that are important from a societal perspective, particularly those related to climate change. Now, worldwide, there are somewhere close to 400 million herbarium specimens, and these are housed in about 3,400 herbaria. And these specimens have been collected for centuries. They go back in time, some of them very old, some of them very recent, but each specimen has a story to tell. Each specimen is packed with data. And so what I'd like to do this afternoon is spend a few minutes overviewing natural history collections and their digitization. And then I'll talk about how we can repurpose collections for new uses way beyond what they were originally collected for. And then I'll give some examples from our own research in ways that we have been doing this. So worldwide, there are somewhere between 3 and 4 billion natural history specimens. And so here we're stepping back from herbarium specimens alone to consider the rest of the natural history collections. And about half of these are in the US. There are also about 1,600 or more natural history collections in the US and thousands of collections in the world. Now, when these collections were first made, they were made to document the biota of the natural world. And in fact, most of our collections, most of what we know about the natural history of our planet comes from these collections. But, and these collections continue to provide us with a framework in systematics and taxonomy, as they have been for centuries. But today, these collections are being used for many different purposes. They provide materials, genetics, for genomics, for analyses of chemistry, studies of species interactions, phenology, biogeography, the impacts of climate change, and much more. But until the last few decades or so, most of these specimens have been locked away in cabinets and have been unavailable for general use. They've been primarily accessible only by specialists who could physically travel to those collections. But digitization is changing that. So as our specimens globally are becoming digitized, we are being faced and presented with many more data points than we had in the past. Now, when we think about digitization, we think first of all of the information about the specimen. So for this specimen of a pitcher plant from northern Florida, the specimen record would include the scientific name, the date it was collected, the collector, the location. That would include perhaps information about the country, the state, the county, the specific site, and for more recent collections, the GPS coordinates. The label might also tell us something about the associated species, and there might be some additional information. Now, if the resources for digitizing were quite extensive, it might be that all of this information makes it into a database. More likely, though, is that the database will have the scientific name, the date, and the location, and possibly the collector. But as these records are being digitized, we have, as a community, have been able to database huge amounts of information. But beyond the label information, digitization also includes imaging and various other sorts of data capture that might be related to that specimen. And so for herbarium specimens, which are generally of standard size and generally flat, this means that imaging is quite easy. And now there are accumulated many millions of images of herbarium specimens worldwide. Now, in the US, the organization that coordinates the digitization and databasing of natural history collections is called IDIG-Bio. And you can see the URL there in the title portion of the slide if you'd like to take a look at that later. Currently, IDIG-Bio has over 138 million specimen records and nearly 54 million media records. And of these media records, about 44 million of them are images of herbarium specimens. And about half of the specimen records are also for plants. So there's a lot of information for understanding more about the vegetation of our world in resources like IDIG-Bio. Now in IDIG-Bio, this is a screenshot of the heat map showing where our collections are located. You can see that there's the majority, the densest collections are from the US, but we also have quite a few collections from Brazil, particularly along the East Coast. Now, it's possible to search IDIG-Bio for in a number of different ways. You can search by species, so for example, Acer rubrum, but it's also possible to search by geography or by a combination of different fields. So if we search for Acer rubrum, the red maple, we are returned a map that shows all of those records that have that are georeferenced. And if you click on each point or pass over each point, you get a very small record that tells us a little bit more about this. And then from there, you can actually view the full record and also find out whether or not there is an image and whether that image might be useful for whatever your particular purpose is. So it's possible to browse through the IDIG-Bio resources to find out what sort of information is available. Now, if one actually is interested in downloading a lot of data, then we have an API, an application programming interface that will allow for that to be done very efficiently. Now, one of the great things about herbarium specimens or any museum collection is that if we know the point from which the collection was made, we can actually use that information to find out a lot more about the habitat and the climate of a particular location. So for example, if we look at the point where this specimen was sampled, we can drill down into that location and find out by using other databases what some of the environmental parameters are for that spot. For example, the temperature and precipitation can be obtained for any point on the map from WorldClim, and we can also obtain information on soil type and soil chemistry from soil grids. And there are various other databases that can be used to provide information on other aspects of the environment. Now, IDIG-Bio is just one such data aggregator, and I've listed a few others here. Of course, in Brazil, you have Species Link, and this was actually one of the early aggregators that sort of led the way for others to build similar platforms. And all of these essentially feed into GBIF, the Global Biodiversity Information Facility. But it's often valuable to have somewhat smaller, localized or taxonomically oriented aggregators as well. So when one might be interested in obtaining specimen record or other data from GBIF, it might be worthwhile also to survey other sorts of aggregators for more local or taxonomically specific information, because not everything from all of these other aggregators has actually made it into GBIF at any particular point in time. So we have a lot of information in these data aggregators, particularly GBIF, which includes not only specimen information, but also observations such as those from iNaturalist or eBird and other observatories. And the really amazing thing about all of these specimen records in particular is the unanticipated uses. When these specimens were collected decades or maybe centuries ago, it was anticipated that those would be used to help document the biota of a region and help form the framework for taxonomy and systematics. Little did many of these collectors, or most of these collectors, know that their samples would and specimens would eventually be used as sources of DNA, as mechanisms for studying climate change, and so on. In addition, collections are well poised to help us address a number of societal challenges. And we see specimen records in various types being used to help us monitor the effects of global change on biodiversity. And this would include things like monitoring the loss of biodiversity, monitoring the spread of invasive species, establishing patterns of changes in land use, loss of ecosystem services, and so on. So for example, ecosystem services might include things like maintenance of clean water, stabilizing the soil, providing resistance to floods, pollinators of crops, sources of foods and medicines, beauty, and of course, so much more. And all of these ecosystem services collectively are worth hundreds of billions of dollars annually. But in addition to various monitoring aspects, these collections can help form the foundation for development of conservation strategies. They can help lead to new approaches to food security, and they can help us track emerging pathogens and other factors related to human health. So what I'd like to do today is provide some examples from our own work on how we're using specimens, including the data from their labels and also the images, to help us predict the impact of climate change and how we might be able to therefore mitigate some of these impacts. And here are the three examples that I will present. The first of these is the application of ecological niche models to the study of Florida plants. Now, Florida is highly diverse in its plants. We have over 4,000 species of plants that occur just within our state. And this is quite extensive compared to most of the rest of the US. Only California and Texas and Hawaii actually come very close. So we were interested in modeling the distributions of species of Florida and species of plants from Florida, and then evaluating how we might be able to use this information for predicting the impacts of climate change, and then also helping to develop conservation strategies. And this is work that was pioneered by a former postdoc in our lab, Charlotte Germain Aubry. Now, to do this work, we used the location information from the herbarium labels, and we obtained the environmental data from those specific points, and then used software to model the ranges of each species. And the examples that I'll show here are based on about 1,500 of the plant species of these 4,100 plants that occur in Florida. And we are currently in the process of remodeling all of these. And we very soon will have models for about 3,900 of the 4,100 species. So again, the first step was to model the distribution of species in the present. So we use the information in the label we find a specific spot, we drill into that spot and get the temperature and precipitation data, as well as soil data. And then we use Maxent software to model species niches and then generate distributions. And we can do this for the future as well as the present by using the IPCC greenhouse gas concentration pathways. And here there are predictions for that correspond to 1 degree to almost 4 degrees C rises in temperature over the coming century. We, in our analyses, use the information from the assessment report, the 5th assessment report. There are now data available for and projections available for the 6th assessment report as well. And we're starting to convert some of our models to those at this point. And of course, WorldClim also has these temperature and precipitation values for the future. So let's take a look at a few examples. The first of these is Albigardia ovata, the flat spike sedge. If we model its habitat and then project that onto the state, we find that its optimal habitat is just in the southeastern part of Florida, essentially in the Miami region. Now, that's not a very good place to be a plant, given all of the pavement and concrete and urbanization, but that's where its ideal mix of temperature, precipitation, and soils would be. Now, if we project those habitat characteristics into the future, we can see where the optimal habitat might be in 2050. In 2050, we don't see any warm colors on this map. And in fact, there is really no optimal habitat left for this species in Florida in 2050. So let's take a look at another species. This is a prunus species, the scrub plum. It occurs in the central part of Florida in areas that are hot and dry, and that's where the model essentially places it. But in the future, as conditions become hotter and drier, it will have suitable habitat in that is much more widespread than the current situation. So we see just by looking at these two species, we have some plants that are likely to be able to survive climate change just fine, and others that will not do well under a hotter and drier climate. Now, when we layer all of the species models onto a map for the present, we get what we see here, the areas of green showing high species diversity, tan showing the low species diversity. And then when we take a look at what this will look like, what the changes will be in 2050, we see green showing areas of increased species diversity, tan areas of lower species diversity. And here we see, as might be expected, higher or increased aspects of species diversity in the north, as we expect that plants will be tracking northward if they're able throughout the coming century. But we also see that some species may be moving southward into the area that is currently the Everglades here in South Florida. So there are some patterns that seem quite predictable, and others that may be a little unusual, and those are worth following up on a little bit. So all of this means that we have some additional tools as we consider the question, what to protect. So this map shows biodiversity hotspots within the US. A few of these, such as the one here in Florida, throughout this region actually of the Southeast, as well as the California Floristic Province and Hawaii, all are part of the list of global biodiversity hotspots. And this information then can help us as we try to think about how to develop conservation strategies. And I'll return to that point very briefly at the end of the talk. So we'll move on next to our second example. This is ecological niche models in agriculture. And this is work that is being led by a current postdoc, Mackenzie Mabry, and some work that was conducted by a former undergraduate, Tori Ford. And our strategy here is that we recognize that herbaria represent huge resources for understanding crop wild relatives and their potential role for future foods crop security. So we can use these herbarium specimens of crop wild relatives to develop ecological niche models and to identify aspects of the niches of these species. And the reason that this is important is that crop wild relatives may provide some of the genetic diversity that might be needed to allow our current crops to persist under models of climate change. And also it may be a way of recognizing which crop wild relatives might themselves be valuable crops in the future. So a herbarium specimen can provide a lot of information for crop-related research projects. So for example, samples of the leaves themselves can be used as sources of DNA, but more relevant to the topics that I've broached here is that we can use the data, the label data, perhaps the images as well, but the label data to help predict responses to climate change of crop wild relatives and of the crops themselves. So we can ask questions about the environmental limits of the crop wild relatives that have already been identified. And we can ask where in the world these crop wild relatives have the highest habitat suitability. And we can do this through the similar sort of niche modeling and species distribution mapping that I just described. So we're in the process of doing some analyses with a range of vegetable crops and their crop wild relatives to identify those crop wild relatives and the locations from which they come that might be particularly useful in crop improvement in the future. We're also able to project where our current crops might be best able to be grown in the future. So again, herbaria are important resources for understanding crop wild relatives and for helping to shape food security efforts. Variation within and among the crop wild relatives can potentially be useful for adapting crops to changing environments. And the niche models and species distribution maps can help us identify crop wild relatives that have either global or very specific local habitat suitability. And we hope that through this process, we may be able to identify variables that contribute the most to helping us understand traits of interest for use in crop improvement. The last example that I'd like to present has to do with phenology and the use of artificial intelligence to understand patterns of phenological change. So phenology refers collectively to those key seasonal changes in plants and animals that occur from year to year. And this would include things like flowering, the emergence of insects, migrations of birds, and so on. And as such, phenological changes are important markers of climate change. In plants, we typically think of bud burst, flowering, and fruiting as the main aspects of phenology, that particularly those that are relevant to studies of climate change. But perhaps as important, or maybe even more important, some of the interactions between species and their either coordinated or not coordinated responses of phenology may be very important. So if a plant is shifting its flowering time, but its pollinator isn't, or vice versa, this mismatch can be extremely severe with regard to the future of this interacting species pair. And the same thing could be true with regard to a plant and its dispersers. So how do we gain information about phenology? Well, one approach that is taking place in the US, and there are similar phenological projects elsewhere in the world, is through a combination of monitoring of target species, satellite imagery, and also community science contributions. And these aspects all go into the data that are reported by the US National Phenology Network. In this image, you can see the darker colors at the bottom, so across Florida, Texas, and the southwest of the US. These are areas where flowering started earlier. As we go farther north, we see lighter yellow. These represent areas where flowering has started recently. And in gray, all of the areas where flower has not yet started. And this was taken from March of 2021. But we might wonder, looking at that map, if that's a typical year, or what do we normally see? Well, we can also get a daily index that tells us whether things are flowering early, as shown in red, compared to the past, or later, as they are in some regions here, as shown in blue. So the NPN provides us with some really valuable information on a nearly continental scale, but it's only been going for a little over 40 years, and it only has a certain set of species that it's targeting. Most of these are actually species that can be seen from satellite imagery. And so it doesn't actually take into account a lot of the understory and herbaceous parts of communities. So wouldn't it be great, though, if we could obtain phenological information about more species and over a greater period of time in order to be able to track whether species are flowering earlier, flowering longer, and so on. And basically, that is the message that we get from the National Phenology Network, that many species are starting to flower earlier, they're flowering longer, and so they flower later into the autumn as well. But again, this is for a subset of species and over a fairly limited amount of time. Well, herbarium specimens actually give us the opportunity to track flowering time back farther, in fact, for centuries, and across many thousands of species. Now, of course, this can be quite tedious if we are scoring things like phenology from the information in the label. For example, if this label said tree in full fruit, we could use the date of collection for this red maple, and we could indicate in our data matrix that this individual was in fruit on that date. But it's also possible to get information from the image itself. So we could score this. We could look at this and say, oh, this is in fruit. You don't need to read any text about that. And then there have also been some software packages developed for scoring these sorts of flowering and fruiting. This is Crowd Curio that was developed by Charlie Willis et al. But still, this work is quite tedious. However, we have millions, literally millions of herbarium specimen images. And what if we could use the power of all of those images, perhaps coupled with artificial intelligence, to allow us to explore the information in those herbarium specimen images over longer periods of time and many more species? Well, in fact, multiple groups of scientists around the world are exploring this possibility. So we've been working with a couple of groups of computer scientists to develop deep learning methods for annotating herbarium specimens as to whether they're flowering or fruiting or not reproductive. And our first attempt at this was finished a few years ago. We examined over 160,000 specimens with this algorithm, and we found that the algorithm was 96% accurate in terms of identifying whether a specimen was fertile. It didn't do such a great job at determining whether something was flowering or fruiting, but it could at least determine fertility. So we thought that was a good start. And so we continued onward and conducted another pilot project. So in this particular example, though, we were interested in whether different species with different morphologies might be more or less amenable to this sort of study. So on the left, we have the California poppy. It has individual flowers that are large and easily identifiable by the algorithm. So in this case, it worked very well to say this plant is in flower. Here are all of these examples of flowers. In the center, we have, again, a maple. And here, the algorithm was very effective at identifying fruits. The flowers are all very small and hard to identify, but the algorithm was quite successful at identifying fruits for species like this maple. However, on the right, we have an orchid with these tight clusters of small flowers. And those individual flowers are not detectable by the software that we developed. And so it was impossible to say whether a species like this was actually in flower or not. So depending on the species of a given area or one's focus, you can imagine that there would be varying levels of success in applying this sort of machine learning approach to identifying and scoring specimens for their phenological state. Well, currently, my colleague Rob Gralnick and a group of collaborators is developing something called PhenoBase, which is an attempt to integrate phenological data from observations like iNaturalist and Project Budburst, along with specimen data and also other occurrence data from GBIF and IDIC-Bio, all in the context of the plant phenology ontology. And the goal of the PhenoBase approach is to develop both resources and tools to enable a much more integrated approach to scoring phenological change associated with climate change over the past decades and centuries. So stay tuned for more work on that. And if you're interested in seeing other applications of machine learning to plant biology, particularly using herbarium specimens, I would just direct you to Applications in Plant Sciences, to a double special issue. The June issue of 2020 explored questions of phenology, morphology, herbivory, species identification, and so on, using various types of machine learning. So this sort of wraps up my main examples, but what I wanted to do was just present you with an overview of a project that we have just started. And this is based on a pilot that we conducted a few years ago, in which we combined information from specimens, herbarium specimens, with a phylogenetic tree for the plants of North America, focusing on the seed plants. And we found here that if we just focus on the right, that the species or the communities in the eastern part of the continent contain species that are more distantly related to each other than expected. And in the western part of the continent, the species are more closely related than expected. So this was a very interesting observation to us. And so we found this pattern of significant phylogenetic clustering across North America in general, but with a difference between the East and the West. We also identified some biodiversity hotspots in California, Florida, Central Mexico, and Mesoamerica, and centers of endemism in Mexico, the southwestern US, and the southeastern US. Now, this was a pilot study that we conducted just to find out what sorts of data are missing, what sorts of tools are missing, and what do we need to do this project better. So finally, after several attempts, we received a grant, which will start in January, to conduct this project. It's called Biodiversity on a Changing Planet. And here we will be focusing on integrating traits, phylogenies, and distributions to forecast risks and resilience of North American plants. And the overall goals here are to try to understand the processes that have yielded current patterns of phylogenetic diversity, current patterns of trait diversity, and how those patterns may be related to specific regions or specific lineages. And our goal here is to be able to identify regions and taxa that are particularly resilient and those that may be under severe risk, so that they can become the focus for conservation planning. And this is a project in which Stephen Smith is the lead PI, and this is being conducted with Julie Allen, Doug Soltis, Rob Gralnick, Brent Mishler, and Sean Laffin. And with that, I'd like to wrap up here and just provide a lot of acknowledgements. I presented the products of a lot of team efforts, sort of as if they were my own, but of course they weren't. And here you can see all of the contributors to these various projects. So I would certainly like to thank all of them, and also thank the National Science Foundation for financial support. And also thank you very much again for the invitation to speak with you, and thank you for your attention. And I know you have a reception coming up, so maybe everybody is eager to go have a drink and some food. But I would be happy to answer questions if there's time, but if not, I know we're running a little late.
Okay, thank you very much, Professor Pamela, for your inspiring presentation. And now, if you allow, we will open for questions, okay? Bom, perguntas.
Hi, Pamela. I'm Patricia Morelato, and I thank you very much for your choice of the choice of your talk. For me, is very important, and I think it's very inspiring for our center as well. And I work with phenology, maybe you know that, and so I think you give me a present giving this talk, personal present. And I like very much, you enlightened a lot of aspects of herbarium collection. I was wondering if you have, I have been working a bit with herbarium collection and phenology, and one thing that we realize, at least for the neotropics, for the south here, is that some of the collection has some we have some ancient collections, like in the early 18th century, 17th maybe, the great collectors. And then we have a huge gap, and somehow mislead us to think that we could do this long time series that does not really exist. And on the other side, we have some difficulty with the labels. So there is a lot of also some misleading in labels, the identification. And so there is, at what we supposed to be an easy task, it is difficult. And sometimes something that we think we are going to reconstruct it is not that much easy. Have you found this kind of difficulties in your work? And also, of course, we did a map of Brazil for the collection. We used more or less 6 million records of Brazilian data on herbarium, okay? And we saw that mostly of our collection, unfortunately, are around big roads, close to most population cities. So, in fact, how much we know of our biodiversity? So this is 3 bias that I see, and I would like to have your opinion on that, if not too much. Thank you so much again for your coming and share your knowledge and all this wonderful work with us.
Thank you. Thanks for raising those really, really important points. Yes, because I was trying to be enthusiastic about the use of specimens, I didn't talk about all of the problems, but you raised a number of problems, the types of biases that our collections have. So as you mentioned, a lot of our collections are very spread out in time in weird ways. They might be constantly collected, but there might be pulses. So there were a lot of things collected at one point and then not again for another long time, and then not again, maybe not even again recently. And so that's definitely a problem. The locations of the collections can be biased by, where people can actually access the areas. So there are some very nice papers, for example, especially by Barnabas Daru, on some of the biases that are associated with herbarium collections in particular. But a lot of these actually are true for other types of collections. And then, of course, we have problems with taxonomy. And so people have, there might be just errors in the label information, but there could also just be changes in the concept, the taxonomic concepts that are used. And so there's really a lot of work that has to go into cleaning the data and sort of filtering out problematic specimen records. So incidentally, I have one of my PhD students, she'll be graduating in a month, actually less than a month now. She is from Campinas, and she went to Unicamp. And so she's been working on a project that's focused in the campos rupestres. And so in order to do that work, she decided she needed to straighten out some taxonomy. And she spent a year working through the information in HaFlora and matching, actually probably more than a year, matching up the species names from HaFlora with a number of other resources. And she's done an excellent job of working through taxonomy. And hopefully, well, she'll be submitting that work for publication soon. But I think that work just illustrates that even from an excellent resource like HaFlora, having that information match up with the data from other collections is extremely challenging. So like even matching that up with data from other collections in neighboring countries, is very complicated. So basically, I don't have like some really good advice other than just to say that these are definitely important challenges, and we need to be very careful about the types of biases that the data inherently have, but in many cases, what the biases in the data may not be related to what it is that you're studying. So the fact that the collections come from along roads, does that actually matter if what we're interested in is changes in flowering through time? So some of the biases may not actually be biases that will bias your future studies. So, I think sorting through all of that it's really important to do that. And so, like, the data cleaning process itself is massive. Sorry, that was a very long answer, but it was a long question, a complicated question, and a very important question. So thanks.
Thank you. Mais perguntas? Se alguém quiser perguntar em português, tá? A gente traduz, não é problema. Nenhuma?
And how do you think, Pamela, it's me again, about to choose some flagship species that are easy to identify and have like big flowers or a very large distribution? I think this could be a way to go around this problem, there's some bias that you have discussed just a little bit with me, but maybe we could get a lot of information. Like we were trying, like we have this floras identified, but also cleaning the data, which was a lot of work. But we also thought about some species like in Brazil, the Ipê, the Randoanthus or Tabebuia, it's like big yellow flowers and are very well identified. We have even we did also a filter by excellent botanists that are good identifiers, good collectors, great collectors, and that we can rely on their identification, something like this we use a bit of artificial intelligence to connect to this. But what do you think about this flagship species? Because I think we could have very nice questions on climatic change and shifts in flooring using not a lot of species, maybe some common species, easy to identify, and then we can have some good data over time. What do you think about that?
Yeah, I think that's a really good idea. In some cases, maybe having a small number of species with really good data could be very useful in helping us identify some baseline patterns that we could then start testing with additional species and additional data down the road. So yeah, I think that's a really good idea. The other thing is that we could maybe test our models. I mean, our whole strategy and approach, and our predictions. So by using things where we know we're not worrying about taxonomy, we're not worrying about collection bias, we're not worrying about some of these other aspects, and we can see how the results for those particular species support our predictions, and that then might help us either accept our approaches or maybe modify them as we go along, so that if species that have good data still give sort of problematic results, then maybe we need to make further modifications. So yeah, I think those are some really good ideas. Thanks for sharing those.
Thank you so much. It was very good. We could talk for hours, I guess.
Yeah,
but anyway,
I think so.
in another opportunity, I'm sure. Thank you so much.
Mais alguma pergunta? Professor Pamela, I have one question, please, about the citizen science. You have discussed about iNaturalist in your presentation, and how is indeed the importance of this kind of database, and how is the bias in this kind of database? Can you discuss it, please? Thank you.
Yeah, so, and was the question about citizen science data?
Yep.
Yeah, so, so I mentioned that actually there's an element of that in all of those different projects, really. So the data that go into IDIG-Bio or GBIF, some of that is, generated through observations from citizen science or community science projects. The same thing is true with some of the scoring for phenology. So, so those can all be sort of impacted by whatever biases there are. And interestingly, people are doing some nice work now, I think, to try to identify biases with regard to not just the collections, but also the observations. So the problem or the bias that was mentioned before about the collections being mostly along roads, well, that problem is even more so when we deal with the citizen science projects. So easily accessible locations. So there are a lot more data points for those sorts of locations, which might end up providing, greater confidence in data that are from those locations than elsewhere. But I think a lot of these biases in the citizen science data really have not been examined that carefully yet, and there are a lot of opportunities. One of our students just finished a study on, well, a lot of people are concerned about the quality of identification of observations, for example, from iNaturalist and other citizen science projects. And so a student of ours did a study where she compared the identifications of some iNaturalist, of images from iNaturalist with images from Herbaria. And essentially there was no difference in the quality of the identifications. So once the iNaturalist observations reach research grade, they are just as likely to be correct as most of the herbarium specimens. So that was a reassuring analysis, I think. But yeah, a lot of the biases in the citizen science data or the effects of the citizen science data have not yet been examined. So there are a lot of opportunities for research there.
Thank you. There is a question here from YouTube from Thais Emilio. Please, Pamela, have you used the AI to extract qualitative and morphological data like leaf or fruit size from vouchers? If so, what do you think are the opportunities and challenges?
So just to make sure I understood the question, so this has to do with with using morphological data taken from specimens or vouchers?
Yes, morphological, like size, for instance, morphometric data, I think.
Yeah, so there are increasingly tools being developed to enable extraction of trait information from images of specimens. So there's a really nice set of software packages developed by Will Weaver and Stephen Smith called Leaf Machine and Leaf Machine 2. And they now have another one that is just becoming available called Voucher Vision. And these all provide opportunities for identifying specific traits on an image and then scoring them. And the Leaf Machine one in particular is very well suited to identifying leaves and actually then doing measurements like length, width, maybe other information, and then putting that information directly into a matrix so that one could do morphometric analyses on there. So there are definitely some things in the works along these lines. And I guess the other thing I could say is that people are taking increasing advantage of the large language models like ChatGPT, although who knows what the future of something like that is right now, given the last few days of upheaval. But using those large language models and natural language processing to take information out of species descriptions. So using the textual information to extract traits. And that's something people have been trying that, for several years now, probably a decade or so. But it's getting easier, and the databases that are being developed are becoming more useful for addressing a bunch of different questions. So I think there are just increasing opportunities along these lines.
Thank you very much. Obrigado, Thais Emílio, pela pergunta. Mais perguntas? No? Podemos encerrar, então? Well, Professor Pamela, thank you very much again for your presence. We are very happy to have you here, and hoping next time you'll be here in person, please. Okay.
That would be lovely. Thank you.
Pamela, I also, as a coordination of the center, I would like to, again, to thank you very much for your talk. Invite you again to come, okay? And also thank you because you were a member of the board that is going to evaluate our center, okay? But it's an honor to have you here and have your talk and chat with you. I hope we can do that again soon. Okay.
Yeah,
thank you so much.
Thank you.
Thank you.
Okay.